Hi everyone! My pathogen for the Human Pathogen Awareness Initiative is Streptococcus pyogenes, or also known as Group A Streptococcus. It is a prokaryotic bacterium, which means that it has a thick cell wall that the crystal violet dye used for the gram stain will adhere to. So Streptococcus pyogenes will stain purple in order to view it under the microscope and also to differentiate from gram-negative bacteria. Streptococcus pyogenes is a spherical, cockeye-shaped microorganism that is arranged singly or in chains, and its metabolic pathway is fermentation. Its optimum growth temperature is 37 degrees Celsius, which makes sense because it is inhabited in our bodies as a member of the normal microbiota in the pharynx. So how does Streptococcus pyogenes evade the body's defenses? Well, it has certain traits called virulence factors, which enhance its ability to cause diseases. For Streptococcus pyogenes, these include extracellular enzymes, anti-phagocytic factors, and toxins. Enzymes allow the microorganism to avoid body defenses and dissolve chemical structures in the body to maintain infection. Hyaluronidase is an example of an extracellular enzyme that Streptococcus pyogenes uses to maintain infection. It breaks down the hyaluronic acid that holds animal cells together, allowing it to invade tissues deeper. Streptokinase breaks down blood clots, which helps Streptococcus pyogenes invade the body through the skin where there is an open wound. And finally, deoxyribonucleases depolymerize DNA released from dead cells in abscesses, reducing the firmness of pus surrounding the bacteria and therefore allowing Streptococcus pyogenes to spread through infected and damaged tissues more rapidly and easily. S. pyogenes produces streptolysins to help it evade phagocytosis from the body's immune system. These membrane-bound proteins lyse erythrocytes, leukocytes, and thrombocytes. So after Streptococcus pyogenes is phagocytized, it releases the streptolysins in the cytoplasm of the phagocyte, causing the lysosomes to release their contents and lysing the phagocyte. So as a result, S. pyogenes is released. Streptococcus pyogenes evades phagocytosis also with M protein, another membrane protein, and by enclosing itself in a hyaluronic acid capsule to help camouflage it, because hyaluronic acid is a normal chemical in the body, so the body will not recognize it. Finally, the last virulence factor is toxins, which Streptococcus pyogenes secretes to contribute to its virulence. Pyrogenic toxins stimulate macrophages and helper T cells to release cytokines, which stimulate a fever, rash, and shock. Now that you have a little bit of background on Streptococcus pyogenes, as well as an understanding of the traits that help it or enhance it to cause disease, I will share with you how Streptococcus pyogenes is transmitted. There are 3 ways through which it can be transmitted. These include direct contact, indirect contact, and through respiratory droplets. Direct transmission involves body-to-body contact, such as touching, intercourse, kissing, biting, scratching, feces to mouth, and across the placenta. Feces to mouth is the portal of exit to portal of entry contact, and across the placenta involves when the infected mother may transfer the pathogens to a fetus across the placenta. Indirect contact involves the transmission of a fomite, which is an inanimate object. A fomite could be a number of things, but a few examples include a needle, toothbrush, diaper, money, bed sheets, medical equipment, and toys. Finally, droplet transmission is spreading pathogens less than 1 meter within droplets of mucus coming from coughing, breathing, or sneezing. If it is more than 1 meter, it would be airborne transmission, which is not how Streptococcus pyogenes is transmitted. It would be less than 1 meter in the droplets of mucus. So because Streptococcus pyogenes is transmitted via droplet transmission less than 1 meter, it is often spread in crowded places such as daycares and schools. Washing your hands is the most effective way to prevent any disease, so wash your hands, often especially when coming into contact with others with fomites or with others who are sick who may have exposed you to their respiratory droplets containing S. pyogenes. Group A Streptococcus causes a number of diseases, but one that you'll be most familiar with is pharyngitis, also known as strep throat. So when you have a fever and a headache and you just feel yucky, and you also have those red swollen lymph nodes and those purulent abscesses covering your tonsils, you may have strep throat. S. pyogenes also causes scarlet fever, pyoderma, which are pus-producing lesions that occur on exposed skin of face, arms, and legs. It may cause streptococcal toxic shock syndrome, which is usually after spreading from an initial site of infection in an immunocompromised individual, necrotizing fasciitis, rheumatic fever, and glomerulonephritis. So this picture here is of an open mouth showing the purulent abscesses covering the tonsils that would be indicative of strep throat. The picture next to it is necrotizing fasciitis, or also known as flesh-eating bacteria, which is when S. pyogenes secretes enzymes and toxins that destroy the tissues and may require surgery to remove the non-viable tissue and the infecting bacteria. Signs and symptoms of Streptococcus pyogenes listed here correspond to the diseases I just mentioned. Remember that signs are objective and meaning observed by others, and symptoms are subjective, which means that they are felt by the patient. So signs include fever, which can be measured by the thermometer, red swollen lymph nodes, the purulent abscesses, diffuse rash, the strawberry red tongue, sloughing of skin, pus-producing lesions on the face, arms, and legs, hypertension, decreased urine output. Symptoms could be headache, sore throat, pain, chills, malaise, nausea, vomiting, diarrhea. Diagnosing Streptococcus pyogenes as the cause of a disease involves the rapid stress test, which is the immunological test used to identify the presence of group A streptococcal antigens. A respiratory sample to detect the presence of gram-positive bacteria is simply not enough because Streptococcus pyogenes, as I mentioned before, is a member of the normal microbiota of the pharynx, so the antigens must be identified. However, if the presence of Streptococcus pyogenes was from a skin sample, where it is not a member of the normal microbiota, just the presence of gram-positive bacteria would serve as a preliminary diagnosis of one of the skin diseases that I mentioned that S. pyogenes causes. Finally, the treatment for an infection caused by S. pyogenes includes the uses of antibiotics. Penicillin is effective against Streptococcus pyogenes, but for those who are sensitive to penicillin, cephalosporin and erythromycin may be used. In addition, bacitracin is a topical antimicrobial effective against S. pyogenes on the skin. And as I mentioned before, surgery may be required in a case of necrotizing fasciitis. It is important to know that Streptococcus pyogenes frequently infects the pharynx or skin, and that it only causes disease when normal competing microbiota are depleted, when adaptive immunity is impaired, such as in an immunocompromised individual, or when there's a large inoculum allowing it to gain an advantage over the antibodies that would form against them. Group A Streptococcus is sensitive to antimicrobial drugs, but it still affects thousands of Americans annually. So remember to wash your hands.